Hello students. In this video we'll study the problems and solution associated with tissue culture. The tissue culture it is an advanced technique to produce the plants rapidly in a mass irrespective of their growing season. It's a technical tool that requires some level of practical knowledge and skill. As we know that the tissue culture technique should be carried out under aseptic condition. So to carry out this, there are some problems that encounter more often if the experiment is not properly done and maintaining the complete aseptic conditions. The most common challenge that we face in our tissue culture lab includes the contamination, vitrification, browning of the media, recalcitrance, somoclonal variation, and loss of totipotency. So these are the problems we encounter during the tissue culture technique. We'll study one by one. The first problem which is associated with the tissue culture it is contamination. Tissue culture requires a completely sterilized or aseptic environment, but any negligence in maintaining the proper environment may lead to contamination of culture, maybe with bacteria, virus, or fungi. It may affect the time, money, effort that we have invested into the culturing of plants. The most common microorganism affecting cultures include fungi, bacteria, molds, yeast. In some cases, they are introduced from outside source, but in others they may be residing inside the associated with explant tissue or entophytes. See, you can see the contaminated culture bottles where the initiation of shoot. See here below in the media, there are molds or fungus which are grown luxuriously. So this is the first problem. How to overcome this? It can be avoided by completely maintaining or improving our sterilization techniques. The room should be clean and the culture space should be organized, equipped with the laminar flow and sterilized with alcohol before working. If we are repeatedly getting, we should improve all this. The consumables, culture vessels, media, and water should be sterilized. The explant should be properly surface sterilized and use plant preservative mixture like products to prevent the contamination from the cultured plants. The second problem, vitrification. Vitrification means it's a physiological disturbance or malformation occurring in tissue culture plants that affecting their proper growth and development. In this case, the plants have glossy appearance and thick, enlarged, and brittle leaves and stems. The main causes of vitrification are the triggering oxidative stress, high salt concentration, high relative humidity, low light intensity, and the gas accumulation in the atmosphere of the jar, and the length of time intervals between subculture and the number of subculture concentration and type of gelling agent, and also the type of explants used may be the cause of vitrification. How to overcome this? It can be overcome by increasing carbohydrate levels in the media and changing the light intensity and modifying the concentration of agar and reduce the humidity in the culture container. In some cases, an increase in the calcium concentration has been observed to reduce this vitrification problem. The next problem associated with tissue culture it is browning of media and explants. Tissue browning is the result of accumulation and oxidation of phenolic compounds in culture tissues. It reduces the rate of cell division and regeneration capacity of explants, but the cause either their death or that is zero response in culture media. This phenomena can be majorly observed in the woody plant tissue. It's also because, as I mentioned earlier, the browning of the media is mainly because of accumulation of phenolic compounds. It can be overcome by using the activated charcoal in the media, where it absorbs the toxins to avoid the accumulation of inhibitory phenolic compounds. And use of antioxidants like citric acid antioxidants in the media to prevent the oxidation of phenolic compounds. The subculture of explants after a period of time to avoid the accumulation of browning compounds, and use of sharp edge knife to cut the explants with and keep them in water for a while, which act as a best solvent for secondary compounds present in the tissues. In some cases, explants placed in the vertical positions also help against the browning of the media, browning conditions, so it can be controlled. Next is recalcitrance. It is defined as inability of plant cells or tissues to respond and in vitro culture environment. The tissue recalcitrance is generated because of the reaction of free radicals with the macromolecules of cells causing the cellular dysfunction. This can be overcome. See, these are all the photographs of the callus initiation. Okay. Uh, the overcome it is done by the selected juvenile parts of the plants as explant and rejuvenate parts of the desired plants using the cytokinin spray of tissue culture. But these are all the healthy ones, what I shown here. It is an explant and it is the undifferentiated mass of cells where shoot induction. Okay, and again the shoot induction in a large amount, and this is the again the callus picture. Okay, and recalcitrance it is can be overcome by juvenile parts of the plants as an explant and rejuvenate the parts of desired plants using cytokinin spray for tissue culture. Next problem is somoclonal variation. It is a phenotypic or genotypic changes in the culture plant. It occurs due to the change in the chromosome structure, number, and DNA sequence. This phenomena is called as a genetic heterogeneity. Somoclonal variation means it's a culture tissue where uh, from the callus or from the explant where you can induce the formation of shoots. But here, in a bulk of all these plant sets, there are few plants which shows the variations. Okay, so these somoclonal variants, somo means a vegetative cell. The clonal means they are the clones of the same cell, and these somoclonal variation because of the chromosomal instability because of the in vitro condition. To avoid that the somoclonal variation, so we have to avoid the long term cultures and to prevent the use of 2,4-D in a large concentration and use auxiliary shoot induction system to prevent this. And propagate chimeras means they are the combination of the two genetic material are the two different DNA sequences by other clonal systems and keep the number of subculturing of plants at a minimum and reinitiate the culture from new explants. The last next problem is acclimatization. Acclimatization is the adaptability of tissue culture plants when brought into the natural environment or in the greenhouse. The plant cultured in a completely artificial environment go under shock and die if not acclimatized properly because from the tissue culture condition from that environment they have to adjust themselves to the surrounding or when they are when we leave it in the field they have to adapt themselves to the prevailing conditions. So these are the hardening of the plants in the nurseries. And the prevention is optimize the hardening condition of the plants for such as mineral nutrition in the media and establish photoautotropic system. Gradually adapt the plants to ex vitro condition, that is from in vitro to ex vitro conditions, like gradually adjusting the percentage of humidity for in vitro plants when transferred to ex vitro condition. These are the main problems and solution associated with tissue culture. Okay. Mm. Apart from this, which are the steps we can take to prevent the cell culture contamination? We have to use the sterile labware. Apart from reagents, you must also sterilize the labware that cells will be in contact with. Use filter tips and change them often, and check your cells often. Plant cells and bleach your contaminated samples, and use good labeling practice and the common sense. In conclusion, the plant tissue culture offers many advantages over the traditional methods of propagation, and it provides a way to fulfill the ever increasing global demand for food resource, and it helps in conservation of endangered plant species while allowing us to make completely a new and beneficial species. So, when we are investing lot on the plant in vitro cultivation of the plant cells, we have to take a suitable measures to avoid the problems associated with this. Thank you.